Hello everyone, welcome back to our channel. So basically in this video we will be discussing about the immunological memory. So what exactly this immunological memory is and how actually B cell memory plays an important role in building the immune memory for different and various pathogens that we encounter in our daily lives. So coming to this, there is one amazing review recently that has been written by the Shane Crotty. So this paper actually explains vaccines, they actually save millions of lives. But how exactly vaccines save millions of lives? Because of the immune memory. So basically when we see the how actually our immune system plays an important role in recognizing the pathogens, the different antigens, and how they actually respond to them. So, first of all, when there is the primary exposure to the pathogen, so the primary immune system, this will take time to respond to that pathogen. So it will take around 7 to 14 days to actually produce the antibodies, while the short-lived plasma cells, they will produce the antibodies. And when there is a second exposure to the same pathogen, how actually it will respond to that pathogen. So there will be abrupt response, the higher response, where memory B cells that have already been there, they will differentiate into plasma cells and they will produce the antibodies, and the titer of the antibodies will be very higher at this level. And so we will be having the higher response to that particular pathogen. The immediate effect that comes through the secondary immune response, where we have already the memory B cells that will respond to that particular pathogen. So in our immune system, when we see our immune system, we have the layered tissue and circulating immune memory in protective immunity. So our protective immunity involves the layered tissue as well as circulating immune memory. So here in case of the tissue immunity, when you see the different tissues, so there will be the presence of antibodies, the CD4 T cells, CD8 T cells, and B cells. And as well as in case of the circulating immunity, the circulating immune system, this will also have the antibodies, memory B cells, and memory T cells. So in case of the healthy tissue, all those layers will be present. If one or two layers are breached by the pathogen, still there are various different types of the layers through which the protection is provided to the particular pathogen. Whereas when there is a severity in case of the infection, so all the layers will be breached by that pathogen and the infection will be very high, and that will cause the immune system to weaken more as compared to the normal healthy tissue. So when it comes to the B cell memory, so basically when we look into the B cell memory, so first of all, if the antigen exposure happens to the B cells, so B cells will actually uh, interact with that antigen through the B cell receptor, and this is called as the antigen mediated activation. And also there will be the naive CD4 T cells, so they will actually respond to the antigen via the antigen presenting cell like the dendritic cell through the MHC class 2, and there will be the antigen mediated activation. B cell activation happens, and these T follicular helper cells, they will actually help the B cell to proliferate in a prolific manner. So B cell proliferation happens, and after that, when the B cell proliferation happens, some of these B cells, they will migrate to the uh, to the germinal center of that lymphoid organ. So when they reach at the germinal center, what basically happens? So there are two different zones in the germinal center. One is called as dark zone, another is called as the light zone. So here in the dark zone, the clonal expansion will take place and B cell BCR diversification will happen. So the somatic hypermutation happens while the different differences will take place and clonal expansion will happen. And in case of the light zone, there happens the affinity maturation where the selection of that uh, cells take place via the when the antigen is presented by the follicular dendritic cell to the B cells, and those B cells which have a higher affinity, they will be selected, whereas those B cells that have least affinity, they will be they will undergo the apoptosis. So here the germinal center dependent humoral memory will take place. So memory cells will be formed, and this will be called as the germinal center dependent humoral memory. And as well as some of the cells from here, they will get converted into memory B cells, and that will be the germinal center independent humoral memory, where they will actually produce the short-lived plasma cells, and these short-lived plasma cells, they will actually uh, they will produce the primary immune response. They will act as an effector B cells, and those B cells will basically eliminate the pathogen that has actually uh, encountered with the host cells. So this will be called as a germinal center independent humoral memory, and there will be certain cells, the memory B cells, that will be called as the long-lived plasma B cells. So they will create the serological memory because they will reach to the bone marrow while they will actually uh, maintain the basic immunity. So they will maintain the baseline of the immunity. And here we have the germinal center dependent humoral memory, where based on this germinal center maturation, affinity maturation, and clonal expansion, so these memory cells will be formed. So we have basically here three different criteria. First is there are short-lived plasma cells. They will act shortly, and they will actually remove the pathogen via the primary immune response. And there will be some cells that will undergo inside the bone marrow, where they will be called as long-lived plasma cells, and they will produce the baseline of the memory. The antibodies will be produced there, and there, in case of the germinal center, where the germinal center dependent humoral memory will take place, and they will also make the memory B cells. We know that, for example, if we have the naive B cells, so this naive B cell, this is the activation of the naive B cell happens via when the antigen is actually uh, the antigen is actually uh, encounters with the particular cell, the B cell, via the B cell receptor, and the CD4 cells, they will actually help the B cell activation. And there will be these plasma blast cells, they will be short-lived B memory cells. And from here inside the germinal center, while the clonal expansion will happen, and then affinity maturation takes place. So there we will have some cells will be long-lived plasma B cells. So they will be called as the long-lived memory B cells, and there will be some cells that are short-lived memory B cells. So they will shortly respond to the antigen exposure. And there are also B memory cells that reach to the bone marrow, where they actually maintain the base level of the immune protection. So as well as here, you can see that this is the for example the resting memory cells. So these are the B cells that are B memory cells, long-lived B memory cells, and after that second exposure, they will instantly undergo the activation, and they will basically produce the antibodies, and the titer of the antibodies will be very high.